welcome everyone today for joining us. My name is Wei, and I will be the moderator for today's presentation. Today's webinar is titled Nanobody Lead Optimization with Epitope Mapping and Binning, Alpaca Antibody Showdown. Before we begin, I will just go over some housekeeping tips. We will be doing a Q&A session towards the end, so if you have any questions, we welcome you to type it into the Q&A chat below. As well, the presentation is being recorded and the recording will be made available to everyone afterwards. Next slide, please. Today's presentation will be given by two of Rapid Novore's senior scientists, Dr. Marko Jovic and Dr. Dominik Narang. Marko leads Rapid Novore's antibody kinetics team with over 15 years of experience in antibody optimization and development. Prior to joining Rapid Novore, Marko held leadership positions at the NIH and in the biotech industry at Nicoya Life Sciences. Dominik leads our antibody sequencing and HDX-MS antibody characterization teams with over 7 years of experience in mass spectrometry. Before joining Rapid Novore, Dominik worked in various academic and industrial settings, including the University of California, San Diego, York University, and Sanofi Pasteur in Toronto. Without further ado, I will pass it over to Marko to begin the presentation.
Oh, thank you, Wei, for that kind introduction. And I would like to thank everyone for joining us today, whether you're watching this live or if you're watching this recording. Also, thank you for taking time to join us that way. So we will kick things off with an introduction to nanobodies. It's a very unique antibody platform that generated a lot of excitement in recent years. And we'll go over some of the specific steps of nanobody development pipeline and how our unique de novo sequencing platform can really supercharge this development effort, and also how particular biophysical techniques such as surface plasmon resonance and HDX-MS fit into that pipeline. So we'll go over kinetic characterization of nanobodies, epitope binning by surface plasmon resonance, and then I'll hand things over to Dominik to go over epitope mapping and also the synergy that exists between these different approaches and how that can really lead to a more accurate, faster, and really more relevant type of leads, taking you to the preclinical stage faster than possible with the standard type of approach. And really, the nanobody story starts back in the late 80s with serendipitous discovery of several students in Dr. Hammer's lab back in Belgium, where they observed that a camelid family produces not only conventional IgGs, but also produces heavy chain only immunoglobulins that are around 95 kilodalton. So this really quickly led to explosive growth of this field in terms of studying heavy chain only antibodies, and also quickly led to development of various constructs derived from the camelid antibodies. And really the one that is leading the way is a VHH recombinant domain. So it's the variable domain of the heavy chain only immunoglobulin, commonly referred to as nanobody, which is a 15 kilodalton fragment. And it has really, given its biophysical properties, it became, one of the really focal points of antibody engineering and development in the last decade or so. Some of the primary characteristics of nanobody that are making it unique in terms of various applications are the thermostability and solubility that are observed with this type of small construct that are very high compared to conventional immunoglobulins. Also, they do exhibit high affinity and specificity. One of the limitations, though, is a short half-life in serum. So in circulation on their own, nanobodies can be cleared on the order of minutes to hours, but a lot of advancements have been made in recent years through various conjugations to make nanobodies more stable through addition of PEG, albumin, FC, whether it's human FC domain or other FC. So really, there are many different ways that nanobodies can be engineered to increase their serum half-life from minutes to hours or days, 2 to 3 days in many cases. Also, as we look at biochemical properties of nanobodies, it's important to point out that they do have a low immunogenicity and a deep tissue penetration. So collectively, that makes nanobodies really a unique platform for antibody engineering. And essentially, this have opened up new research perspectives in development of innovative human therapeutics. In terms of application, really, some of the most relevant features are the extremely long CDR3 loop that is found that is unique to nanobodies. So this is nanobodies derived from any camelid species, whether it's alpaca, llama, or camels. And with this CDR3 loop, the nanobody is capable of really reaching deep into various binding cavities, so really opening up epitope repertoire that was previously not accessible by standard antibodies. Also, due to their small size, they exhibit better stability and solubility, so they're more tolerant to acidic conditions, temperature fluctuation, and as a result, also that translates to ease of engineering and manufacturing on a large scale. What is really relevant and exciting is actually their ability to penetrate tissues deeper than any conventional immunoglobulin. So in oncology, there are already some very promising and exciting applications to delivery of various payloads deep into tumor tissue, and also in the field of neurobiology and delivery of various drugs across the blood-brain barrier. The fact that nanobodies, many of them naturally and some of them through some engineering efforts, can be designed to effectively cross the blood-brain barrier, it holds a huge promise in terms of the future applications. Now, one of the limitations in the field of nanobody development for a long time has been the process itself of designing nanobodies, whether it's the traditional phage display that comes in many different flavors, or more recent efforts that are based on various NGS-driven approaches, so sequencing approaches in various cellular systems, whether it's bacteria, yeast, or human cells. They commonly have same limitation, which is the availability of B cells in circulation that results in clones ultimately that are not most physiologically relevant, and they're not necessarily top affinity binders. And essentially sequences that are obtained for the nanobodies through these efforts many times do not directly reflect the immune repertoire in terms of the way that they're represented in the living organism. Also, this has implications for downstream development, because many of those leads, even if they have high affinity, are because they might not be most physiologically relevant and they're more engineered constructs, they might exhibit issues in terms of toxicity, stability, aggregation. So in PK studies or downstream development, issues might come up with those types of leads. So that is why we have developed really a new approach, a unique approach to obtaining antibody sequences through direct de novo sequencing in combination with machine learning. So what we'll be sharing today is the first successful sequencing of alpaca immunoglobulin proteome de novo with a technology that directly sequenced IgGs from the immune repertoire of alpaca. And as a result, we were able to identify more physiologically relevant, high affinity binders while not being limited by the availability of B cells in circulation. Now, we do have a lot of resources on this platform itself. So we have actually a couple of webinars, as well as blog posts, and really many different types of resources on our website if you're interested in specific steps of how these samples are processed and different types of species that can be used for our repap platform analysis. But really, in a nutshell, for this particular webinar, what I would like to point out is essentially through these efforts, we are able to obtain sequences of heavy chain only, as well as regular immunoglobulins coming from alpaca immune repertoire, and those sequences can then be used downstream for nanobody synthesis efforts in development. In terms of the pipeline where this technology fits in, so this is the in the discovery phase, of course. Once we have those sequences from the repap platform, we can move quickly down the funnel through screening, whether it's based on surface plasma resonance, ELISA, or some other way of screening, and quickly identify top leads, top candidates that can be used for full kinetic analysis, obtaining on rates, off rates, KD, and moving along, then also studying those top leads in concert. So looking at their compatibility in terms of epitope diversity through epitope binning by surface plasma resonance, and also epitope mapping using HDX-MS. In simplest possible terms, really, what we'll be focusing on today and what's the real power of the synergy between our repap platform for de novo sequencing of polyclonal antibodies and HDX-MS and the SPR is the ability to obtain the nanobody sequences quickly from this immune repertoire, and then answer two key questions, which are how and where these nanobody constructs are binding on the respective antigen. So to answer the question of how, in terms of kinetics, we use surface plasma resonance, and to answer the question of where, primarily we are using HDX, although we can also couple it to surface plasma resonance through epitope binning to gain deeper insights into the nature of the epitope-paratope interaction in 3D space. So we'll focus first on characterization of the nanobody hits that came from alpaca repap study. And really to understand that and make sure we are all on the same page, I will just step back for a moment and just go over briefly of the some of the basics of surface plasma resonance technology. So in essence, surface plasma resonance is a biophysical technique that enables us to study interaction between two biomolecules, whether it's protein-protein interactions or lipids, carbohydrates, or small molecules. The surface plasma resonance is a really versatile and accurate platform in studying any kind of biomolecular interactions. The premise is that you have one of the binding partners immobilized onto the gold surface, and we can take advantage of properties of gold in terms of surface plasmon and the evanescent field around the gold surface to detect the binding of the binding partner that is present in the solution. So as we immobilize one binding partner that we refer to as ligand, we can pass over the second binding partner in appropriate buffer and detect the binding. Now, there are different types of surface plasma resonance platforms out there. A traditional SPR instrument such as BIACOR uses planar gold surface that has the evanescent field, so the field of detection of around 250 to 1000 nanometers. And that is the field within which any binding event can be recorded thanks to the laser light source that is shined onto the gold that creates that is reflected at a certain angle, and the angle changes upon binding of the analyte. So essentially, with this type of SPR instrument, we are looking at the change of angle that corresponds directly to the binding event, the molecular weight of the analyte bound. One of the other approaches to the SPR is the localized SPR, and this is the technique we use for studying antibody-antigen interactions. Essentially, rather than using planar gold, the SPR instrument that we are using, OpenSPR from Nicoya Life Sciences, is using 100 nanometer gold nanoparticles. Now, colloidal gold has a much smaller evanescent field of around 20 to 40 nanometers, which translates to much lower background interference coming from the buffer, and essentially any particulate that might be present in the buffer in the standard setup can be detected within the evanescent field, even if it's not actually binding to the immobilized ligand. So we get better signal to noise ratio. Also, as a result, we have reduced bulk shift, and that downstream leads to faster optimization, higher sensitivity. So this really works well for the antibody antigen setup. It might be a little bit more challenging when studying small molecules or really large analytes such as cells. It also uses a little bit of a different approach where LED white light source is used to essentially detect absorbance peak shift upon binding. But essentially, it is a very sensitive way of studying antibody-antigen interactions. So why do we need to use SPR in antibody discovery and in particular in development? Well, it gives us deeper insights into the kinetic profiles of each binding pair, and it does so with very high accuracy. It's really the gold standard of antibody development. There are different ways that SPR can be used in the discovery and development pipeline. It can be used as early as during the screening process, identifying those top hits among hundreds of antibodies. And it can be, it is most commonly used when characterizing top hits and developing those leads down the funnel. Essentially, it is a very versatile platform that also can be then further used for antibody engineering efforts. As we introduce modifications, mutations in our top leads, we can reassess kinetic profiles of those modified antibodies and optimize for a particular application. Also, it's very useful in quality control and batch-to-batch validation. In simplest possible terms, to visualize what SPR can do, we can look at this diagram on the left. And here we're looking at, imagine that you have several leads coming from the initial discovery phase that all have the same affinity, or let's say 100 nanomolar. That would be pretty bad. Antibody, we can say it could be a 1 nanomolar or 100 picomolar, but really the concept is that ultimately with the affinity alone, it will be very difficult to make a conclusive pick on the top lead for further development. And the reason is because really the affinity is only telling us a part of the story, because with this here we're looking at clones that could have the same affinity but would have very different on and off rates. So if we compare this analyte shown in yellow, it is sort of middle of the range, still having the same KD, but having average on rate and the average off rate. Now, if you look at the a black trace here, it has a very fast on rate. Once the analyte binds, it remains bound until we switch back to the running buffer and the analyte comes off. So the off rate is very fast. So this interplay between the ratio between the off rate and on rate, both being very high, results in the average KD. And the opposite is true of this light blue trace, where we have very slow on rate, but also very slow off rate. So it really, with SPR, it can give you this type of rich kinetic information set that can drive the rest of the development effort, depending on the application. Do you need ideally an antibody that is having a very fast on rate, or maybe you don't care about the on rate? It's the off rate that really matters. So the way that for all of these types of analysis, the way we would approach a standard SPR experiment is by immobilizing one of the binding partners onto the gold surface and then passing the analyte in solution in increasing concentrations. So initially, as the analyte is introduced, for the first little while during the association phase, what we will be observing is only binding of these analytes onto the immobilized ligand. And as the association phase progresses, what will start kicking in is the dissociation as well. So we will get into from the first order reaction to second order reaction, basically having the interplay between the on rate and the off rate until we reach this equilibrium here at this particular concentration of the analyte. And then once we switch back the flow to buffer only, so just using the running buffer, what will happen is as we wash away the analyte, we will get to the scenario where we're in the dissociation phase, where the only thing we'll be recording is the off rate, the KD. And actually, when we do the analysis of SPR sensorgrams, the mathematical model starts with the off rate as a first order reaction, then it factors it in for calculation of the on rate. Now, ultimately, for especially for the multi-cycle kinetic analysis, in standard SPR experiment, also we need to figure out regeneration conditions to remove any analyte that might still be bound at the end of the dissociation phase. In terms of antibody development, then if you look at our alpaca nanobody set, so the way we used SPR is essentially we took the ligand that was, so for our ligand we used the antigen that was used to immunize alpacas. So in this case it was a rabbit antibody. So we mobilized the rabbit monoclonal onto the carboxyl sensor. So this is gold functionalized with carboxyl. And what we're looking at on the bottom is essentially an optimized immobilization setup for the nanobody study by SPR. There are a couple of steps that go into the ideal immobilization conditions. So figuring out immobilization is not a trivial matter in order to get just the right balance of the ligand concentration on the sensor that will give you most accurate kinetic readouts. So in this case, using carboxyl sensor, we activated carboxyl groups with EDC NHS. So essentially, we charge the EDC NHS so we can perform, this is the green trace here, followed by the yellow trace, which is the injection of our ligand, so our rabbit monoclonal. So that was here we see that we can get a really nice capture onto the sensor surface. And if you follow it up with injection of BSA to block any remaining charged carboxyls, we see that there is very little of that left, but still to make sure that we are quenching the reaction, the amine coupling reaction, what we do is we inject 1 molar tris in this case, but you can use other amine reagents such as ethanolamine. Now, in parallel with this channel here that we refer to as active channel, we can also perform a similar injection sequence in a reference channel. So that's what we have here on the right. So essentially the blue trace is identical to what we are showing here, broken down by injection. So that is the active channel. And then we have a reference channel shown in red. The only difference being that we do not inject our ligand in the active channel. We only inject the BSA, and we get nice immobilization of BSA, saturating the surface and blocking any non-specific binding that might be happening. Now, again, as I mentioned, there are a couple of different factors that go into immobilization optimization, such as the pH. So we need to perform usually pre-concentration study to determine the optimal pH to be used for capturing the ligand on the carboxyl surface. Also, screening the density, that is range, that getting to the right concentration of the ligand, where we avoid ligand packing and potential steric hindrance. So those are all the things that need to be factored in when designing an SPR experiment, and in particular immobilization. So once you have that in hand, you can move to characterization of your analyte. In this case, our nanobodies that came from the de novo sequencing effort. So we picked a couple of top clones here, R1, R2, 4, 7, and 9. And we studied their ability to bind to mobilized ligand by injecting increasing concentrations of each of these analytes. And really, for a good spectra and for appropriate kinetic analysis, what we need is several injections, not just the one injection, but at least 3 to 5 injections. So in a range that spans 10% of the expected KD to up to several fold the KD. So here for a 1 nanomolar interaction, we're just, for simplicity, we're just showing a couple of traces with fit curves, but spanning the range of 100 picomolar to 100 nanomolar. And there are a couple of ways that we evaluate the accuracy of our analysis and accuracy of the kinetic parameters calculated from this kind of set. So we look at model fit. So the optimized system is processed using appropriate binding model. Usually we always want to go with the simplest possible model and without any corrections or heavy processing. There are models out there that include diffusion correction or some of the other artifacts that might be really an experimental problem. It needs to be tackled through experimental design rather than mathematical modeling. So we always recommend just going with the simplest possible model and dealing with any potential issues, whether it's mass transport limitation, any kind of diffusion or dispersion issues, dealing with those experimentally. So once you have a good fit, you can evaluate it by measuring chi-square. So we aim for the 10% cutoff of the Rmax. So chi-square values being less than 10% of the theoretical Rmax that we also calculate to be such where you avoid any potential steric hindrance. And also we measure residuals in a way that they need to be also less than 10% of the highest individual response. Again, looking at essentially the noise distribution, showing that it is indeed random and it's not affecting your data set, not skewing in a particular direction. So in the case of nanobody characterization, what we observed is that with a couple of our top leads coming from the alpaca study, we were seeing some very diverse kinetic profiles. So here we have clones R9 and R4 as our top binders shown in green, R7 is our weaker binder, and then R1 and R2 not binding to immobilized ligand. The R4 and R9 are showing binding that is in the high picomolar range, so represented here by the KD averages, while R7 seems to have a binding more in the nanomolar range. And even just looking at these sensograms, just immediately can see that there's some very unique kinetic properties, with R9 having a relatively fast off rate compared to R4. It has the slowest off rate, represented here by this plateau, with R7 actually having both the relatively fast off rate and overall low KD. So this is something that is very important when determining which nanobody you pick for in a particular downstream development in your engineering efforts. Now, for simplicity, again, we are just showing a couple of clones, but you can imagine this being done with dozens of clones at this stage. Also, I mentioned earlier that nanobody on its own, in terms of application, can have very short half-life. So one of the ways to tackle that is to conjugate it to albumin or human FC. So when you do that, you also have to go back and characterize those derivatives. So those constructs, once they're fused to, for example, human FC, so you get a humanized VHH construct, you still need to go back and characterize it, not just for avidity effects, but overall binding could significantly be different from the nanobody alone. So in this case, what we assayed is VHH, so nanobody fused to human FC. And what we're seeing is that while the KD seems to be in a similar range for R4 and R9, what we are seeing is definitely an increase in binding with R7 that is stabilized by this additional scaffold. And also now at this point, we with the FC present, we also start detecting binding from R1, which really brings us back to this idea of assaying de novo most physiologically relevant clones, because doing this kind of kinetic study with nanobodies, you could miss out on R1, where actually that might be, for a particular application, a really important lead, right? So, and it does seem in this kind of stabilized conformation is the humanized VHH, as HCAB, it does have a pretty healthy KD of 41 nanomolar. Now, looking at this kind of kinetic analysis can be a little bit confusing and challenging. It can be a challenge to really tease apart like some of the trends that might be happening, especially if you're looking for a very particular kinetic profile. So maybe you're looking for an off rate in a certain range or an on rate in a given range. So to better understand that and really get some deeper insights from kinetic sets, what we do is we can also do isofinity analysis, where we can compare the dissociation rate here plotted on the x-axis against the association rate. And these types of analysis can be very useful when you're looking at a large number of leads. So here, just an example, we can see that for R4 and R9, we're seeing very different trends. They seem to have overall a KD that's represented by diagonal lines. Again, this KD is representing the dissociation rate divided by the association rate. So that's the diagonal. So what we see is that with R4 and R9, they are sort of clustering together, although we're seeing the trend that the nanobody for the R9 has a much faster off rate that's slowed down by introduction of human FC. With R7, it's we see a different trend. So with actually introduction of FC leads to slower, with the R7 we're seeing the slower off rate, while with R9 we're seeing the slower on rate. And also we see the appearance here of R1 with introduction of FC. Now that doesn't really tell us the whole story. The kinetics is just part of the puzzle. So just to give you a flavor of how SPR can really give you some of the structural data, we can also take a look at the epitope binning, essentially looking at assaying the epitope diversity of these nanobodies. So with the epitope binning, what we can do is essentially we can look at how these antibodies can bind to their respective epitopes in combination, in pair, right? So this is done by injecting one antibody at saturating concentration, followed quickly by injection of a second antibody, and then assaying the response of the second antibody in the presence of saturating antibody compared to the response you get like with that second antibody on its own. So this is what the injection sequence would look like. Here we have each of the clones injected individually compared to the injection that is done in sequence, with R9 being the saturating antibody. So for example, with R7 and R4, we'll be comparing this type of response to the original response of each antibody on its own, or nanobody in this case. So what we are seeing are some really interesting distinct patterns of epitope diversity. Again, for simplicity, we're just showing 4 clones. And with this type of study, what we want to look at is essentially this distribution of epitopes in individual bins, where any kind of inhibitory activity between 2 clones would place them in the same bin, meaning that those 2 antibodies or nanobodies are not compatible. So you would, if you're using, if you need 2 nanobodies for a particular application, you would probably start with R7 being in the single bin, and then you can assume that it's safe to combine it with one of these other clones. If you need R9 or R4, so you would have to choose one and then pick another nanobody from a separate bin. But it also collectively, this gives us some hints of the epitope diversity and the structural complexity that might be driving these type of binning results. We also observed it with humanized HCABs, so the presence of FC does affect binning, but again, it doesn't tell us the whole picture. So in order to really understand what is happening in 3D, what is happening on the level, structural level of that epitope-paratope interaction, what we need to do is we need to perform HDX-MS and really do, dive deeper as far as structural information of those interactions. So I'll hand it over to Dominic to go over the HDX-MS part of the study.
Hello, everyone. Good morning, everyone. I'm Dominic, and I'll talk about the epitope mapping of antibodies sequenced using our repap platform and characterized using SPR kinetic analysis. Just a second, sorry about that. So what is epitope mapping and why it is important to study epitope mapping? So epitope mapping is a process to define the binding of an antibody to an antigen. So an antigen can be a protein molecule or a sugar molecule or an antibody. In our case study also, our antigen is an antibody. It is important to understand the mechanism of antibody binding to the antigen so that you can understand how the antibody works. And moreover, once you know where the antibody is binding on the antigen, you can select complementary antibodies to improve the antibody response or to improve the efficacy of the antibody. And in addition, if you know where the antibody is binding on the antigen, it will help to protect your patent rights as well. So there are multiple techniques available for doing epitope mapping, and the gold standard is X-ray co-crystallography. If successful, this technique can provide single amino acid resolution with a high degree of confidence, but you need a very high amount of protein sample to begin with, and you need a good luck to start with. And the next is mutagenesis. So this is again a high resolution technique, but again, it's a labor intensive and it takes a lot of time to create different mutants. And the third one is a peptide scanning. So peptide scanning, in this technique, you basically synthesize a pair of a pair of overlapping peptides so that you can test those peptides against the antigen antibody complex. So if that particular peptide is able to disrupt the antigen antibody interaction, that could be a potential epitope. But again, in that case, it's only useful for linear epitopes. And then the last one is a hydrogen deuterium exchange. So HDX is a relatively fast analytical technique, and you will require only a small amount of protein as compared to the X-ray co-crystallography. And you can do screening for 4 to 5 maps in a single day, which is relatively faster as compared to the other techniques. So now coming to the theory for hydrogen deuterium exchange mass spec. So as the name suggests, it's an exchange of hydrogen atoms with the deuterium. And since deuterium is 1 Dalton heavier than the hydrogen atom, high resolution mass specs can easily distinguish between the hydrogen and deuterium. So there are 2 important factors which really affects this exchange rate. One is the solvent accessibility, and the second is the hydrogen bonding. So the hydrogen atoms which are residing at the surface of the antigen, they will exchange much faster as compared to the hydrogen atoms which are in the core of the protein. Second, same is true for the hydrogen bonding, like hydrogen atoms which are part of the alpha helix or beta sheet, they're gonna exchange much faster as compared to the hydrogen atoms which are present on the intrinsically disordered regions. So looking at types of hydrogen in protein, so hydrogen atoms in protein are either covalently linked to the carbon atoms or to the functional side groups like hydroxyl group and the amide backbone. So the hydrogen atoms, which are covalently linked to the carbon, they do not exchange during the course of our experiment. And similarly, the hydrogen atoms, which are part of the functional groups like side chain, hydroxyl, they exchange so quickly that we don't detect those changes in our experiment. And the amide hydrogen exchange slowly on the time scale, and the exchange of these amide hydrogens are monitored using hydrogen deuterium exchange mass spec. So HTXMS is a 5-step, doing an HTXMS experiment is a 5-step process. So the first step is to incubate the protein in D2O buffer for certain intervals. And after that, we quench the reaction by reducing the pH to 2.5 and the temperature to 0 degrees Celsius. After we stop this reaction, we digest the protein using online pepsin column or other low pH digestion columns. And after we digest the protein into peptides, we separate them using C18 analytical column. And after we separate them, we detect them using a mass spectrometer. So this is our platform. So we use LEAP HTX platform for preparing the sample. The first step, which is deuterium exchange, happens in this particular chamber. And the quenching happens at the second chamber, which is maintained at 0 degrees Celsius. And then we have a pepsin column at the back, and then we have a C18 columns in this chiller, which keeps the temperature at 0 degree. And then we use water cyclic ion mobility mass spec to detect the peptides. So before going into the results, I'll discuss about a sample report. So this is a sequence coverage map for the proteins. So this is the first set of experiments, and each solid line over here represents a peptide. So I try to get like near to 100% coverage for the protein of interest. And after we get the coverage, the next set of experiment is to look at deuterium uptake. So we compare the free state of the antigen with the bound state of the antigen to the antibody. And from there, we generate uptake plots. So this uptake plot is basically an uptake of deuterium of the free antigen, which is black, compared with the red one, which is the bound state of the antigen with an antibody. So this is another way of representing the same data. So here it represents the whole amino acid sequence of the protein, and it's called heat map. Heat map is a fractional uptake difference between the free and the bound state of the antigen. If you look at the color scheme, so if there is a decrease in deuterium uptake in the bound state, then you will see a blue. And if there is an increase in deuterium uptake in the bound state of the antigen, then you will see something on the red side. And if there is no change, you will see a green color over here. Similarly, we can map these changes on the crystal structure or a homology model to look at the 3-dimensional epitope for that particular antibody. So now coming to results, as Marco mentioned that our antigen is an antibody, so we will have 2 different sequence coverage map. One is for the heavy chain, and the second one is for the light chain. So once we get the coverage map, the next step is to look at the deuteration of these particular nanobodies. For the HTX epitope mapping, so I use the FC fused version of nanobodies and use these following nanobodies for epitope mapping. So I will go through the results in the same order. So I will start with R9, followed by R4, R7, and R1. So now coming to R9, so this is the heat map for the R9, and this is a heat map for the heavy chain of the R9 bound to the antigen. So here we can see that I only observed decrease in deuterium uptake in this particular region for the R9 when it is bound to the antigen, and the decrease is quite significant over here. And the uptake plot also showed a significant decrease in deuterium uptake as compared to the free one. Next, we map these changes on the model structure. So map is shown in gray cartoon, and the region which showed difference greater than 4% are color coded, and the difference which is less than 4% between the free and the bound state are in the gray color. So in this particular case of R9, we just observed changes in this particular peptide here, and it is next to an unlinked glycosylation site. So it's very interesting that this site is next to an unlinked glycosylation site, and it's not very usual to have an epitope site which is next to a conserved unlinked glycosylation site. So the next one is the R4. So on the R4, seems like it's binding on the C terminal of the Fc region as compared to the R9, which was binding on the constant region of Fc. So this R4 is binding at the C terminal, and along with the primary interaction, seems like there is another interaction which is happening is in the Fab region of the antigen. So seems like this is also involved in the interaction of this R4 with the antigen. So now coming to R7. So in case of R7, I only observed changes in the heavy chain of the antigen, and and those changes are only observed in the early time points. So it's very clear from this homology model structure. I observed changes only at the 10-minute time point. Here we can see that there is a decrease in deuterium uptake in the Fab region, but at 60-minute time point, the decrease is not significant. So suggesting that somehow the map, the R7 map is removing from the antigen, and it's letting the exchange of hydrogen with the deuterium. And the last one is the R1, which is the weakest. So I only observed an increase in the deuterium uptake in the light chain of the R1, which is very unusual as compared to the rest of 3 maps. And this particular region on the model structure is solvent exposed, and it's part of the Fab region. It is very interesting. So if I put together all the four epitopes, so seems like the R1 and R7 are binding on the Fab region of the map, and R9 and R4 are binding on the Fc region of the map. If we revisit the Binning experiment, here we can see that R4 binding is partially blocked when R1, R7, and R9 is bound to the antigen, which is very interesting. It is true for all other antibodies, but vice versa is not true. For example, when R4 is bound, it's not affecting the binding of any other antibody. So that's very interesting. So why it is like that? So if we look at the R4 binding, seems like R4 is binding over here, but its binding is affected by the binding of R9, R7, and R1. One potential reason could be that the binding of R4 requires both interaction with this region as well as the Fab region. So when R9 is present, or R7 is present, or R1 is present, so it somehow blocks its interaction with the Fab region of the antigen and partially blocks its interaction. Whereas on the other way around, when R4 is bound to the Fc region, it doesn't affect the binding of R9, R7, and R1. So which is very interesting. So it's kind of like in this scenario, you need both epitope mapping information as well as binning information to really understand why R4 is partially blocked as compared to other nanobodies. So these are some of the key insights from our work. So using our polyclonal sequencing platform and SPR kinetic experiment, we were able to identify 4 different maps. 2 of those maps bind to the Fc region of the antigen, and they are high affinity. And two other antibodies are binding in the Fab region of the antigen. And R1 and R7 are interesting in the sense they are binding on the Fab region, and these R4 and R9 are binding on the Fab region, and these 2 are binding on the Fab region of the antigen. So by combining these sophisticated experimental techniques, we can identify novel antibodies against any antigen of interest. So our platform is able to work with any antigen, and we can identify novel antibodies for that target protein of interest.
Great. So I'll take us into the outro and the Q&A. So thank you, Dominic and Marco, for another wonderful presentation. Before I turn it over to the Q&A, I will leave you with a few quick words about us here at Rapid Novore. Since our inception in 2015, we've had the pleasure and experience of successfully completing over 7,000 proteomics projects and are rapidly approaching the 8,000 project milestone. We are Canada's largest privately funded mass spec proteomics facility, regularly providing our de novo antibody sequencing and characterization services to many of our global clients, which includes 18 of the top 20 pharmaceutical companies in the world. Our growing team of 65 dedicated scientists and experts comprises world leaders in bioinformatics, protein chemistry, and mass spec proteomics. Next slide. I would like to invite everyone to also visit us at rapidnovore.com to explore some of the other great services we have to offer to help with your projects. These include our ReMap de novo monoclonal antibody sequencing, RePab de novo polyclonal antibody sequencing and antibody discovery, MatchMap peptide mapping, as well as rapid SPR kinetic profiling, rapid HDX-MS epitope mapping, paratope mapping, and ligand binding services, which were highlighted in today's presentation. And finally, we have many online resources available for you to explore and to learn more about the science and applications of SPR and HDX-MS. These include our scientific article blog, past webinars, publications, and case studies. If you had any additional questions following this webinar, you can always reach out to Marco and Dominic directly, and they will be happy to get back to you. We've included their emails here, and we'll leave this slide up while we go through the Q&As. Or you can submit your questions to info@rapidnovore.com. So thank you, everyone. And I'll jump into a few of the questions here at the chat. The first one is, if the antibody is more than 95% purity, can we map the sequence by a tandem mass spec? I think this one's for Marco.
Yeah, so so we can definitely do that. And actually, this sounds like that would be a monoclonal antibody, which is really, our core platform, that is the ReMap platform. So we'll be happy to discuss that project further. But yeah, it's sounds like a perfect candidate for a ReMap analysis.
Okay. Next question is, can you screen a nanobody library using SPR?
Yeah, so definitely. Actually, SPR is really, a great fit for screening dozens and dozens of nanobodies. So early in the discovery phase, using the SPR to assay kinetic profiles of initial hits is really, I would say, in my opinion, I'm of course biased, is the right way to go. We can usually, what's done in that case is you pick basically a KD that is an acceptable KD that's basically a cutoff, let's say 100 nanomolar, a really high, a high KD that would identify all of the relevant hits. And then we would assay those against the immobilized antigen in a way that we would look at response levels immediately post injection. So immediately as the dissociation phase starts, we would collect a reading and we would look at basically what we refer to as complex stability. It's a good, representation, like it's a rough representation, but it's a good representation of overall affinity. It doesn't give you really an accurate, number in terms of the KD, but it is very effective in screening those high affinity binders versus average affinity binders and non-binders and low affinity binders. It's actually also more appropriate than using ELISA, because really the challenge with ELISA is great in determining protein concentration. It can be used for affinity analysis, but to do a proper affinity analysis by ELISA, what you need to know is the off rates. You really need to have those kinetic parameters to determine your incubation times with ELISA. And based on the off rate, you find that with many of those potential hits, you might need 10 to 15 to 20-hour incubation to get accurate affinity measurements by ELISA. So actually, SPR is probably the right tool to use even early in the process, so screening nanobodies. And what we can also do is usually when we do our screening projects, we not only look at complex stability, but also we measure off rates and then make a combined ranking between those two parameters. Because sometimes you have, a high affinity clone that has a very fast off rate. So it might be, sort of just written off, like it might not be, on top of the list, given the very fast off rate.
Great. Next question is, what would the effect of high glycosylation in this SPR and HDX MS workflow?
So in terms of SPR, so high glycosylation, it really depends on the orientation. So really the assay setup. If you have highly glycosylated analyte, it can be a challenge. It can be very sticky, as many SPR labs discovered. So some of the highly glycosylated proteins don't really work well with SPR. Now, with that being said, in theory, it shouldn't really matter what the orientation is for the two binding partners with SPR. In reality, that's not always the case, but you can immobilize the highly glycosylated protein, use it as a ligand, and then pass the binding partner as an analyte that might not be as sticky. So yeah, that does need to be factored in when designing an experiment. And maybe Dominic can speak to the HDX aspect.
For the HDX, it would be difficult to identify those peptides due to the nature of that glycan, because generally glycan structure, unlinked glycans are very complex in nature, and identifying those peptides because of their heterogeneity is difficult. So we might lose like 4 to 5 amino acids around that glycan side in that information. Otherwise, it's not a problem in a regular workflow.
Right. Next question is, maybe this was already answered. Can you do the epitope mapping when the antigen is a glycan?
No, we cannot do that.
Okay. Next question, the VHH, for VHH sequencing to identify high avidity VHH, do we provide you the llama serum? How long will you get us the sequences and how many sequences? What affinity range can you get?
Yeah, so that is a question for repap team, so for our polyclonal sequencing team. So just based on the general description, I would say, I mean, it really, with the repap platform, it really varies from, project to project. So I would urge you to just connect with us. If you can fill out just a quick form, we can get back to you today and set up a meeting. But yeah, so basically you would have to provide, the llama serum, and it's really, the question is, what is, your need for that project? In terms of affinity, it's, I mean, it really depends on the llama. So depending on the immune response, how, and then also what is important there is, having enough of that antigen that we can use for characterization. So doing full kinetic analysis, maybe HDX-MS, all that.
Great. So just I think we have time for just a couple more questions. Next one is, I have an non-immunoglobulin antibody that has 10 binding sites. If I want to do SPR-based kinetic experiments, how can I reduce the avidity component interfering with my assay?
Yeah, that's an important question. So the way to do that is essentially, there is a little bit of optimization that would go into that type of setup, where essentially when you have a multivalent situation with the 10 binding sites, you have to, in simplest possible terms, you have to do an avidity analysis. So you have to essentially find the concentration of the ligand where you go so low in the concentration that the spacing of the ligand is such that they're so far apart that the only binding that happened between the analyte and immobilized ligand is in the monovalent scenario. So there are ways that you can confirm that. So we talked about chi-square, about residuals. So essentially, you want to decrease the ligand concentration that would essentially increase the spacing, and then having that monovalent binding, you would perform, increasing injections of the analyte. Really, still, like, once you have that, the mobilization part optimized, the rest of the analysis would be done the same way. But yeah, it can be done. It's just a little bit more of a custom project.
Okay, 2 more questions here. For the epitope mapping and binning, how many antibodies can you do?
Yeah, so for binning, it's I would say usually up to a dozen. Anything beyond that is just I mean, it can be done. It's just the question is like, is that necessary? Just in my experience, I would say if you're looking at more than 10 to 12, you might have to go back and sort of reanalyze or weed out couple of those leads. But I mean, it's just a matter of, it's more of a matter of study design.
I think the same is true for epitope mapping. We can do 10, 20 antibodies against the same antigen.
And the last question we'll have time for today is, do you have a capture method for nanobodies as a ligand for SPR binding kinetics?
Yeah, so we have actually good in-house antibodies, anti-VHH antibodies that work really well. And we have those actually optimized in the sense that we have a set of regeneration conditions that pretty much work for any VHH, any nanobody. So we would immobilize, covalently couple anti-VHH antibody onto a carboxyl sensor, and then we would capture a nanobody that would be used in this case as a ligand, right? We would pass over it an antigen as an analyte in a single concentration, and then essentially to do this right, we would have to regenerate the whole thing. So strip off nanobody with analyte bound and then remobilize, right? So it does take a little bit more time, but it's really, it works really well. It gives you very good, dose-dependent responses that are very accurate, but it does have that extra step of capture of the nanobody as a ligand and then regeneration of the whole thing and rerunning it with a higher analyte concentration.
All right, so I think I'll wrap it up here as we are up at time. If we didn't get to your question today, please feel free to reach out to Dominic or Marco directly, and they'll be happy to get back to you promptly. So again, thank you everyone for joining us today, and have a good day. Take care.
Thanks, everyone.